Hey all, welcome to ShareTreck. This is Raj here. Friends, today I'm going to talk about a very foundational topic that is very important for genomics investors. At least I believe so. And this is my worldview on gene editing technology and its evolution, as well as the current state of play of licensing of CRISPR-Cas9 and the direction in which gene therapy is going, why it is unaffordable right now and why it could be more affordable in future and probably revolutionize the face of medicine as we know it. So for all these details, please watch the video until the end. And of course, as always, I love your feedback and your opinions. So please do not forget to mention that in the comment section. That said, let's get started. Welcome back, friends. As a genomics investor, we keep on trawling the net to find information about the status of various gene therapies in the clinical trials. So that's one area of activity that we have on an ongoing basis. The other is about who owns the license, because CRISPR-Cas9 license is a complex labyrinth at this point of time. And if you could recollect that when Casgevy got approved, promptly Vertex went and bought CRISPR-Cas9 license from Editas, even though CRISPR Therapeutics had already granted CRISPR-Cas9 license to Vertex for creating Casgevy. This indicates that you can't take a lackadaisical approach towards CRISPR-Cas9 licensing, and if and when your therapy gets approved, then it needs to have a proper coverage of license from both parties, CVC as well as Broad Institute. And of course, after that, there have been a lot of other Cas proteins that have been found with very specific properties, creating a veritable toolkit for gene editing, with each Cas molecule being suitable for a particular aspect. And of course, we have got our delivery technology like liquid nanoparticles and AVVs, adenovirus vectors, and lentiviral vectors, and so on, which are also trying to improve their ability to penetrate to various cell types, and also their ability to carry a bigger payload. And of course, along with all these things, we are addressing the issue of double strand breaks and the locations on the genome where double strand breaks can be dangerous. And also we are creating new technologies where a scaffolding or a kind of template is provided so that after the break the reconstruction can be done properly. We have new technologies like base and prime editing, which can accomplish the task of gene editing without double strand breaks. So there's a whole lot of exciting things happening, and this video is going to present the way I visualize gene therapy and its licensing and its usage, and how it's going to transform medicine and its uptake from being a very expensive technology to being something that most people will be able to afford. So the way I look at it is that gene therapy represents a transformative approach to treating genetic diseases and protein defect related conditions, but it's important to recognize that it's not a cure-all. However, the progress we have made in this field is remarkable and offers hope for addressing a wide range of disorders that were once thought to be untreatable. Our journey began with technologies like zinc finger nucleases or ZFNs and TALENs or transcription activator like effector nucleases. These early tools allowed for precise gene editing, but they were complex, time-consuming, and costly to design and implement. Although ZFN and TALENs could be used for in vivo editing, their practical applications were limited due to the difficulty of tailoring them for specific targets. The advent of CRISPR-Cas9 marked a significant leap forward. This revolutionary technology simplified the gene editing process by using guide RNA to direct the Cas9 enzyme to specific DNA sequences, making it far easier to set up and operate compared to ZFNs and TALENs. The introduction of base editing and prime editing further refined the process, allowing for even more precise modifications without the need for double-strand breaks in the DNA. These advancements have opened up new possibilities for correcting point mutations and making subtle changes to the genome with greater accuracy and fewer off-target effects. Despite these technological strides, the cost of gene therapy remains high. The complexity of these therapies, coupled with the rigorous regulatory environment, has kept prices elevated. However, as technology continues to evolve and as regulators like FDA become more familiar and comfortable with these tools, we can expect the costs of gene therapies to decrease. This in turn will lead to wider acceptance and accessibility, potentially ushering in a new era in medicines where genetic diseases and some protein defect related conditions can be addressed more effectively. Licensing landscape of CRISPR-Cas9 technology has evolved very rapidly. The rapid evolution of gene editing technologies has also led to a complex and evolving intellectual property landscape, particularly around CRISPR-Cas9. If you are using CRISPR/Cas9 in the US, a broad license may not be sufficient due to the ongoing patent disputes. The Broad Institute and CVC, which is the consortium between the University of California, the University of Vienna, and Emmanuel Charpentier, have been embroiled in a high-profile patent interference case, and primarily concerning the use of CRISPR-Cas9 in eukaryotic cells. The US Patent Trial and Appellate Board, or PTAB, initiated interference between CVC and the Broad Institute is currently under appeal, but this interference only applies to certain CVC applications related to eukaryotic cells. Despite this, CVC holds more than 55 patents in the US covering CRISPR-Cas9 use in all cell types, including eukaryotic cells, and these foundational patents remain unaffected by the interference action. Therefore, if you're operating in the US, you need a CVC license to use CRISPR-Cas9 technology, and you might also need Broad Institute license if your work specifically involves eukaryotic cells. So with this kind of a proposition in place, we as genomic investors, when we are investing in various gene technology companies and following their progress along the FDA clinical trial and approval towards monetization, we have to always remember that at the end of the day, a license has to be bought from both the Broad Institute as well as the CVC in order to be absolutely covered from any kind of litigation. Outside the US, the CRISPR-Cas9 patent situation is much clearer. In the Europe, CVC holds the broadest foundational patents for CRISPR-Cas9, as the Broad Institute initial patents have been revoked or substantially narrowed. CVC's patent cover the use of CRISPR-Cas9 in both cellular and non-cellular settings across major global markets, including Canada, China, Brazil, Australia, and India. With over 100 patents globally, companies planning to use CRISPR-Cas9 are highly likely to require a license from CVC. It's important to note that one license is unlikely not enough due to various components involved in CRISPR-Cas9 technology. From the underlying reagents to the methods of use, CVC's patents cover many aspects of the technology, such as the use of single guide RNA or sgRNA and modifications to transactivating CRISPR RNA or tracer RNA, while ERS Genomics offers a comprehensive package of CVC patents. Additional licenses may be necessary, particularly from Broad Institute, for niche applications or work involving eukaryotic cells. Regardless of the ongoing patent interference case in the US, the foundational patents held by CVC remain robust, and these patents are crucial for using CRISPR-Cas9 in any commercial application, meaning a CVC license is indispensable. And on the other hand, you can also say that the Broad Institute license is also equally indispensable because if you're going to have a global reach for your therapy, you need to be covering all your bases. So all our genomic investments in any of the companies that we have in our watch list, we have to look at where they will have to spend money on getting licenses once their CRISPR-Cas9 based therapy gets approved by FDA for monetization. So in conclusion, I'll say that while gene therapy is not a one-size-fits-all solution, the continued evolution of gene editing technologies like CRISPR, base editing, and prime editing provide immense potential for treating genetic diseases and protein defect-related conditions. As these tools become more refined and as the costs associated with them decrease, gene therapy is poised to become a cornerstone of modern medicine. However, navigating the complex licensing landscape, especially around CRISPR-Cas9, is essential for any entity looking to utilize these groundbreaking technologies. And as investors, we have to watch very carefully that and make sure that our companies where we invest in are covering all their bases with their licenses. And that of course means the cost for getting these licenses, which again translates to the profitability of those therapies. And these licenses are one of the reasons why the cost of these therapies are likely to be higher, because these licenses will have to be monetized within their validity period, and therefore they are slightly higher. And with proper licenses in place, I think the promise of gene therapy can be fully realized. And as investors, we have to be cognizant of the fact that there's a very delicate balance between patient well-being and morality in terms of helping patients overcome adverse health conditions and looking at profitability. So that balance has got to be made, and therefore there will be various actors who will come into play when gene therapy gets monetized. And the balance of all of these things will result in the profitability that we as investors in gene therapy can expect to achieve. So I think we have to be realistic in our expectations of profitability with gene therapies. And over a period of time, all these patents will expire, and it will become generic. And at that point of time, gene medicine will be much inexpensive, and more players will be in the market, and more therapies will be covered. And I think as time goes by, we'll have more patents coming up for very specific genetic tools which are built on top of CRISPR. And they could be nucleases like Cas9 or Cas12 or any of those things that we have already got, but new discoveries. And that will probably fuel even more refinement of and even more accuracy and effectiveness of therapies. And at the same time, we'll also see an evolution of delivery mechanisms, whether it is LNP or it is adeno-associated virus or some other viral mechanism which can carry a higher payload and which can also penetrate a variety of cells and ensure that the therapy reaches all the places that it is supposed to reach. So all those things are happening in parallel. I think that the best of gene therapy is still ahead of us in another decade or so. And in the meantime, our patients as investors in gene therapy is going to be tested because these companies are going to suffer a lot in the high interest rate regime. But in that case also, I'm looking at light at the end of the tunnel, because with a September rate cut coming in the horizon, the mindset of the market might change, and with easier money available, more venture capital can come and support these startup companies which are working on gene therapies. And that should help us as investors in companies like CRISPR Therapeutics, Beam, Editas, Verve, you name these companies. Even companies like Bluebird Bio might get a little bit of a push up, and I think that should help us in the long run. But at this point of time, I think it's a question of patience for us as investors. So with that, my friends, I would like to end this video here. I hope you got something valuable out of this, and I would look forward to your feedback. Are you going to be a patient investor, and how long do you plan to stay invested in genomic companies? And do you think we need to add any more companies to our watch list of genomic companies? Please mention that in the comment section, and I'll consider them and make changes. Thanks, and have a great day. Bye for now.